Welcome to this session. My name is Renee McCormick, and I'm really glad you're here. I'm the Chief Geek of the National Math and Science Initiative, with science in particular, and this particular unit is arguably the most important unit in all of AP Biology. I would have to put it in second place. I must confess that I do believe, as much as I love chemistry and biology, and thus I love biochemistry, I gotta say evolution ranks supreme in when it comes to the study of AP Biology. It's probably the largest theme running through the entire course, as I hope you've already figured out. But let's get on with biochemistry, and we're going to start with an investigation, an inquiry type investigation, because that's one of the things that your teachers are going to be really emphasizing with you this year in this course, because inquiry is becoming a big theme no matter what AP science course that you're taking, whether it's biology, chemistry, or physics. And speaking of AP chemistry, yeah, I'm that same lady that did the 150 AP chemistry screencasts as well. So whether you are currently an AP Chem, have been an AP Chem, or are going to be a future AP Chem student, you may endure me again. This is a Texas twang, and it's not going to go away. So you may have to rewind and say, what did that crazy lady say, on more than one occasion. So let's carry on and examine our first little sort of quandary. This is an inquiry moment, and they all start with a question. So, what creates the devil's gardens in the rainforest? Well, first of all, what is a devil's garden? It's got a very cool name, because the natives, the locals, actually believed in ancient times in particular, and some of them even in present times, that evil spirits are cultivating this garden, and it's evil spirits that make these bare zones in the most species-rich area on our planet, which is the Amazonian rainforest. Well, scientists, of course, don't particularly believe in evil spirits, so they go ahead and make a plan. And the plan is to set up some experiments, collect some data, analyze the data, and try to formulate an actual explanation of why these little zones with no particular vegetation exist in the most, again, species-rich rainforest on the planet. So we have two players who are actually responsible for this. One of them is this tiny little ant, quite adorable, don't you think? Shumani. I can't say the first name. That's his genus, and this is the species. But Shumani's the best I can do. And then which tree? The Herusta, and again, I may not be saying that right either, but that's the species of the Duroia trees that are particularly involved in this phenomenon. So again, who tends this garden? Well, we need to formulate some hypotheses. We've already discussed the reason why it's called a devil's garden. So the hypothesis number one is that the garden is being cultivated by evil spirits. Well, hypothesis number two might need to be a tad more reasonable, which would be that this could be plant versus plant warfare. And so perhaps the Duroia trees themselves kill competing trees by means of some sort of chemical put into the soil. That phenomenon is called allelopathy. Allelopathy. And it's very common on this particular continent with black walnut trees. Ask around. If you haven't ever noticed this, ask grown-ups. Do you know much about a black walnut tree? You know anything interesting about a black walnut tree? And their answer will most likely be, yeah, you can't get anything to grow in its root zone. The hulls of the black walnut, because you know a walnut is actually originally in a hull, just like a pecan is in a hull on the tree, and then the hull opens, and then perhaps the nut falls to the ground. So the walnut's hull is actually black in color, hence its name, the black walnut. And when it falls to the ground, then it gets wet and it exudes a chemical, leaches a chemical down into the soil, which prevents any other organism, pretty much, a plant organism anyway, from inhabiting its root zone. It's well known for that. But perhaps that's what's going on here. Perhaps the leaves or the tree itself is exuding some chemical into the ground that is inhibiting the growth of vegetation of other species. So that's hypothesis two, plant versus plant. Think of it simply. So what is the logical next step now that you know those ants are involved? Yep, that would be animal versus plant. Perhaps those Shumani ants living in the trees are actually doing something to the ground themselves, or something is taking place with them and the way they are protecting their turf, basically, since that's their food supply. So perhaps it's producing some kind of poisonous chemical that kills trees or shrubbery of other species. So any good scientist, once they've pondered a problem to solve, then goes about to design an experiment. So Deborah Gordon, along with Michael Green, which are graduate students of a professor named Megan Fredrickson, sought the cause of these devil's gardens. And they performed field experiments in Peru. And so you can see that essentially we have this zone outlined here in yellow, this zone where we were going to have the trees themselves, the Doroya trees, and putting some saplings of another species in. But note, there's a difference. One of them has a protected outside kind of ring. It's out, or oh, I'm sorry, this hasn't happened yet. These aren't here yet. This is step one. So this ring right here is what I want to focus on, and that is an insect barrier. So we're going to try to figure out between, and it's not just one plant, you know, there's like bunches of plants, but those that are essentially unprotected versus those that are protected against the insects. So 2 saplings of the non-host tree species were planted inside each of 10 devil's gardens. All right, let's talk about that for a second. Oh, look, not 1, not 2, not 3, but 10 devil's gardens. This speaks to sample size. You are going to most likely be asked at some point during this course or even on the AP Biology exam to design an experiment. You want to mention sample size. It's an easy point to get. Your whole life, because in the interest of time, you have had what I call the Goldilocks phenomenon. Most of the labs you've ever done have involved 3 trials. So you've done, maybe temperature is a variable. So you've done warm, cold, and hot. That's, you know, Goldilocks in the porridge. No. You need many, many more than just 3 trials, many, many more than just 3 gardens here. So they used 10. And again, there was a sticky insect barrier, so literally like a fly trap, piece of fly paper put down sort of, to capture insects and keep them away from these newly planted cedrela saplings. And the others were left unprotected. Well, why? Again, we want protected against what? Against ants. Or unprotected against ants. So 2 more of these saplings were introduced about 50 meters, which is a good long distance, outside each garden. In other words, what's the point of this? Well, the point of that is to make sure that if the ants are responsible, were the Doroya trees even necessary, or do the Doroya trees have nothing to do with the situation and it's just the ants? So if it were just the ants, this tree would be protected and this tree would suffer. So you have to think through several variables at once. And then to really, really nail it down, they also chemically analyze the contents of the ant's poison glands because, you guessed it, if you take the organism out of the picture, but you extract that chemically, whatever the contents is that they are secreting into this vegetation to kill invading species, then what's the story? Well, it's the chemical alone. The ant just happens to be the one injecting it in. So, many, many trials, many, many plants, many, many ants, and lots of observation, data collected, recording. Eventually you can get to results. So if you look at this bar graph, this is dead leaf tissue in centimeters squared area after 1 day. Ooh, fast acting. Very fast acting in the land of biology. 1 day? Really? The largest amount of dead leaf tissue were on the saplings they had put into this devil's garden that were inside and unprotected. If they were protected and inside, hardly any of the dead tissue. So that means that both of these are in the same environment inside the zone of those Doroya trees, but this one had no ants, or very little ants. I won't say no, I'll say very little. Very little ant impact. Outside, notice it didn't matter. So this rules out this idea that the Doroya trees themselves are not not part of the problem, which means they are part of the problem. So it's a combination of things. So this ant, what's taking place is this ant has made injections from the tips of their abdomen with this particular chemical into the leaves of the unprotected saplings in their garden, and within one day those leaves develop a lot of dead area. So look at that graph again. The ants of this species, the Shumani species, kill non-host trees by injecting the leaves with a chemical, and they even isolated that chemical and know it as formic acid. Now, if you live in the part of the U.S. or anywhere else where there are fire ants, there's also formic acid in ant bites, and it's the formic acid that sets you on fire and makes your tissues sting as well. Does the same for plant tissue. It's damaging tissue. So it creates hospitable habitats for the ant colony. So the ant colony's got it pretty good. They're making sure that there are no competitors within their habitat, within their garden, their devil's garden more specifically, so that no other vegetation can grow there and interfere with or compete with their own food supply. So here we show that the ant, which nests in the stems, that's its habitat, creates the devil's gardens by poisoning all of the plants except its host with formic acid. So by killing these other plants, it provides its colonies with, again, abundant nest sites, long-lasting benefit, and they can live for 800 years. These colonies can. No, not the specific ant, the colonies. All right, so what does all of that have to do with the task at hand? Well, you got to understand, again, that biochemistry, in understanding the interaction between an ant and a portion of the rainforest, all comes down to the molecular level. So I'm really hoping you've seen this thing before that's on the screen now, which is the periodic table, and that you know some stuff about it. For instance, hopefully you know how it's arranged. You know that it's called a periodic table because the properties of the elements repeat, yep, here it comes, periodically. And so you should also know that it's arranged by these numbers, and that these numbers are the atomic numbers, which is the number of protons, and that you may even know what some of the zones are called. You may know that these are the transition metals. You may know that these are metalloids, meaning they have properties of both metals and nonmetals. Hydrogen's a freak over here in the list of alkali metals, and these are alkaline earth metals all down this row. And they have different characteristics. So what's the scoop on these sad little, they don't even have a pretty color, do they? These gray coded elements down here at the bottom. Well, those are the lanthanides and the actinides. Lanthanides and actinides. Uranium is the last naturally occurring element on this planet. I know, it was named after Uranus, a planet, and then we have Neptunium, Plutonium, etc., etc., etc. Most of these were named for the Lawrence Laboratory at the University of California, Berkeley, where all of these other elements were man-made, so they're synthetic. And so anything after uranium, including uranium, I should say, are going to definitely be radioactive. Compounds. Compounds exist of two or more of different elements combined in a fixed ratio. We're going to study many, many, many different kinds of compounds as we move along. And a compound has a characteristic different from its element. That's what's unique about it. So they're different from their elemental properties. For instance, your good buddy sodium chloride. When we say salt, there are many, many, many salts on the planet. This is the one that is on your table. So we call it table salt quite often. But sodium looks like this. It is a soft metal, so soft you can cut it with just an ordinary kitchen knife. Now, this person's wearing gloves, because if you know much about sodium, you know that the slightest little piece of sodium put into water makes a big kaboom. Because hydrogen gas is generated as a product of the reaction. Sodium hydroxide is the other product, and the hydrogen gas, and this is a very exothermic reaction, can sometimes ignite. So sometimes you get a really big kaboom when you've got that gas that has been set on fire. Think Hindenburg. Chlorine is a poisonous gas. Note its color. It's kind of a little bit greenish yellow. So chlorine, again, comes as Cl2 in its elemental form, because it's one of the diatomics. All the gens are diatomic: hydrogen, oxygen, nitrogen, and all the halogens. And the halogens reside right here: fluorine, chlorine, bromine, iodine, and astatine. So this poisonous gas, which would kill us if we were breathing it, is they very, very strange, very dangerous properties, yet in combination, they don't harm us at all. We actually enjoy them. So it's a seasoning that we sprinkle all over our food without any fear. So about 20 to, no one's going to ask you this stuff, okay? This is just interesting stuff. Okay, it's interesting to me. About 20 to 25 of the 92 elements are essential to life, and carbon, hydrogen, oxygen, and nitrogen. Carbon, hydrogen, oxygen, nitrogen. Carbon, hydrogen, oxygen, nitrogen make up 96% of living matter. Most of the remaining 4% consist of other things that you've heard of. Calcium, yes, strong bones and teeth. Phosphorus, as in ATP, the P, the phosphorus part. Potassium, another thing that we need. You eat, if you're an athlete, you probably know this. If you have ever worked out and you've gotten leg cramps, especially down here in Texas where it's hot, really hot, and you dehydrate quickly, you can cramp up. And if you've ever watched sports on TV, you'll see an athlete, a gymnast, or a football player or a basketball player cramp up and get cramps. Well, potassium is one of the things that our muscles in particular need, and a good source of potassium is eating bananas. So you may have been told at some point, eat a banana, you'll feel better, and nobody explained to you why. And then sulfur as well. So trace elements are those that are required by an organism in minute quantities. So we'll run into some of those as well. For instance, I bet you can name one. Okay, I'll give you a hint. It's in your blood. Okay, actually, it's in your blood cells. What do you think? Yep, iron. Okay, iron. But it's going to be in ionic form, so it's going to be either iron 2+ or iron 3+. But we need some of these trace elements. So no one's going to ask you to memorize this. This you probably already have an inkling about. Nitrogen, 80% of our atmosphere. Hydrogen and oxygen in water. You know that carbon dioxide is coming out your nose right now. So there's your carbon-oxygen combo. So you probably already have a good feel for this. You know that there are nitrogenous bases in DNA, if you've had a biology course before, or even a life science course in middle school. But these are the ones that are in trace amounts. No need to memorize them. Again, I say, no need to memorize them. When we go through all the rest of the units of the course, you'll understand how each one of these play into a specific piece of the biology that we study, but there's no reason to memorize them. All right, so an atom is the smallest unit of matter that still retains the properties of an element. Classic definition. You probably already know it. An atom is composed of subatomic particles, neutrons, protons, and electrons. The protons and the neutrons reside in a very dense nucleus, and the electrons reside in the electron cloud. Okay, let's talk about that really dense nucleus. I am from Texas, where football is almost a religion. If you can imagine a football stadium, so imagine like your high school football field or a high school football field in general. Maybe not the Dallas Cowboys Stadium, but a stadium where a high school game might be played. The protons and the neutrons that are packed very densely into the nucleus would be like a flea on the 50-yard line between the hash marks. BTWN, between hash marks. The electrons, however, reside in the entire rest of the stadium. Flea, entire stadium. So the electron cloud is not dense at all, and the electrons are just sprinkled throughout. The reason, as you might expect, is you know that opposites attract and likes repel. So these electrons out in this cloud are going to be repelling each other. Also, what you might not know is that the mass of the proton and the neutron is about 2,000, and I say about, okay, it's 1986 if you want to know exactly, but 2,000 times as massive as the electrons. So the electrons are really light little particles, and they move around, and they are constantly out in that electron cloud. They're not always evenly distributed, but they are always repelling each other. So here's a decent little picture, but I have to say right here that I hate this. It is in every textbook on the planet, and I keep thinking one day, I really am going to live long enough that one day we take these out, because this diagram is indeed fraught with danger. This is more reality. there's this area, a cloud, if you prefer, where the electrons are actually in. So we're going to have an electron somewhere in this cloud. One of them might be closer than the other. One of them might be, farther apart. They are going to repel each other, and so if they ever end up together, somebody's moving. Or they may both move, but they're going to spread out as best they can. Now, this implies an orbit, and we even call them orbitals, but unfortunately that terminology stuck like 120 years ago, and we've never been able to get out of it. Originally, we did think they orbited because we think in a planetary model most of the time, the way our solar system works, but lots of experiments in the early 1900s said that is very wrong, but yet we still keep seeing it over and over again. So again, just so you can read test questions, the atoms of various elements differ in number of subatomic particles, and the atomic number is the number of protons. So when we talk about carbon, carbon is always 12. Carbon is not always 12. Can I extract that statement? Can I have a do-over on that? Carbon is always 6. Often has a mass of 12. That's the stable isotope. But we also know that carbon's got two other isotopes. We'll talk about isotopes here in a second, 13 and 14 as well. So an element's mass number is the sum, as in addition, the sum of the protons and the neutrons. And atomic mass is the atom's total mass that can be approximated by the mass number, but it's actually represented by an average based on the fact that it has various isotopes. So what is an isotope? Of course, speaking of isotopes, all of the atoms of an element have the same number of protons. As soon as you change the number of protons, you're not that element anymore. So carbon has 6 protons, nitrogen has 7. So you can't change the number of protons and it still be the same element. But you can have different numbers of neutrons. So isotopes, and there are many I words. You might as well get used to it. I find that students have a real hard time with ion versus isotope versus isomer. So I'm going to make a real big point of getting these solidified in your brain. Isotopes. Those are two atoms of an element, the same element, that differ in the number of neutrons. Differ in the number of neutrons. Coming back to carbon. There's carbon 6, 6, and 6. It has 6 protons. But it can have a mass of 12, which means it has how many neutrons? This is, are you smarter than a 5th grader, Jeff Foxworthy math? Come on. 12 minus 6 would be 6. So it has 6 protons plus the 6 neutrons, and that's going to make the 12. Then there's an isotope of carbon 13. Oh, yes, you're catching on. So that has 7 neutrons because it's going to always have 6 protons, or it's not carbon. And then there's the one that you've probably heard of that we use for radioactive dating, carbon dating, which is not a comment on carbon's affinity for other carbon. But 6 protons, and then 14 minus 6 would be 8 neutrons versus 7 neutrons versus 6 neutrons. So these two are radioactive. This is the normal one. And interestingly, for about the first 20 elements on the periodic table, a 1-to-1 proton to neutron ratio is usually, well, I can even say always. I don't get to say always very often, so let me have that moment. Always more stable. So that first 20 elements. When you get more and more and more electrons in the electron cloud, more and more protons in the nucleus, then more neutrons are necessary because the protons are repelling each other, because like charges repel, and they're all positive. And so we edge up more to like 1 to 1.2, 1 to 1.5, even when you get up to big elements like uranium with almost, 95 to 100 protons in there. You're going to have a whole bunch of neutrons to keep those protons from repelling each other right on out of the nucleus and being unstable. So we already talked about the fact that we use isotopes in carbon dating, but we also use isotopes in some medical research kind of things. Like anyone who has a thyroid issue often undergoes a test where they inject iodine-131 into their body so they can actually watch how the thyroid is processing the iodine, and we can put it under x-ray scopes and things like that and be able to see how it's traveling through and how it's degrading as it's supposed to, and measure those rates and make a diagnosis from that. So we diagnose quite a few medical disorders that way. Iodine-131 is just maybe the one that you've heard about before. All right, so take a moment and see if you can answer this question. Which of the following is the criterion upon which the modern periodic table is organized? Pause me. Go ahead, think. Don't cheat. Pause me. Pause me now. All right, you ready? Made your choice? Indeed, the number of protons. This is the atomic number on that periodic table. Try another. Why might scientists be interested in the atomic mass of an atom? This has more words to read, so pause me. Think about it. Don't cheat. All right. Why might scientists be interested in the atomic mass of an atom? If we know the atomic number, we can determine the number of neutrons. It will not tell us how it behaves in a chemical reaction. It does not let us know the valence of the atom. The valence is the outer electrons that are reactive. We cannot use it to know if the atom is radioactive or not. It doesn't tell us that. But the atomic mass will let us do that simple subtraction and determine the number of neutrons. All right, enough about the nucleus, onto the electron cloud. There are energy levels. This is where we're going to explain what that valence thing really means. Energy is the capacity to cause change, perhaps by doing work. You probably learned in around 3rd grade that energy was the ability to do work. So that is it's a common thing once again in textbooks. This one is actually true. There are many forms of energy. Potential energy and kinetic energy are not the only 2 forms. You're big kids now. You got to learn there's a bunch more forms. But you probably have already learned that potential energy is the energy that matter has because of its location or structure. You learned it as location. And you learned it as location off the ground. So what you've really learned, unless you've had a physics class, you've really been focusing your whole life on gravitational potential energy. Well, there's a lot more than that. That's not the only kind. And chemistry and biology relies heavily on the idea of separation of charge. When we talk about cell respiration and photosynthesis and electron transport system, what drives it is energy. That is a form of potential energy, but it's not gravitational. It's not about how far the mitochondrion is off the ground, because that's silly. It's about the separation of pluses and minuses and separation of charge and how opposites attract. And if you keep them apart, then you're setting up a potential energy situation where they're going to come back together, because that's what they want to do. They want to attract each other. So the electrons of an atom differ in their amounts of potential energy because they're all stuck in a space and they're all negative. And the electron state of potential energy is called its energy level or electron shell. Okay, I'm about to have a little tirade here. I hate the word shell. It is just fraught with misconception, but biologists are determined to keep writing it in textbooks. The chemistry people sometimes write it, but most of them have shifted away from that word because shell, in this language at least, implies a really hard surface, and that is not at all what the edge of an atom is like. It is completely fluid in terms of its ability for those electrons to move around. It doesn't have a defined shape. It's just, if you've ever played with something called oobleck or goop, it's just, they're like that. They've got a little structure to them, but they're not like hard spheres, is what shell kind of implies. So it's fraught with misconception. You're going to see it in test questions, though. So I have to warn you that you're going to see it in test questions, and you need to think of the shell as the electron cloud or the perimeter of a space. Now, how do electrons move? You probably have enough chemistry knowledge to be dangerous and know that they move from energy level to energy level, more specifically orbital to orbital within a sublevel. But it's a quantized event, like quantum energy, like quantum mechanics, Q-U-A-N-T-I-Z-E-D, quantized event. Now, you're familiar with quantized events because you're in high school, and you know that there are a certain number of credits that you need to be classified as a freshman, sophomore, junior, senior, and if you fail a class, you may not have enough credits to start your junior year, let's say, as a junior. You may be classified as a third semester sophomore. This happens in college regularly. And so you have to have a certain number of credits, and not everyone can go to college full time, so it may take them a long time to get to be the next classification. But you can't sorta be a senior. You either are or you are not. And that's what we mean when we say a quantized event. So they use an analogy here of a ball bouncing down a flight of stairs, because you can't be in between. You cannot levitate between this area of that stair and this area of that stair. You're either on this stair or you're on that stair, but you cannot hover somewhere in between. So they like to use this in biology books about an analogy for the energy levels, because the ball can come to rest only on each step, not between the steps. So my language is that's what a quantized event means. I can be here or I can be here, but I cannot be anywhere in between. So how do these changes happen? Well, it's all about energy changes. So the first shell, here we go again. The first shell, which is really not a hard surface, is the lowest energy. So to get to a higher energy, energy has to be absorbed. And so energy has to come in. Well, how can we make an atom absorb energy? You've probably done this. If you've ever done something called a flame test, then you have made an electron, actually millions to billions to gazillions of them, absorb energy. If you put them into a Bunsen burner flame, you'll know they absorbed energy, because you'll see when they come back down. Because what goes up in energy is eventually going to come back down to ground state. When it comes back down to ground state and comes back this direction, that's too far, comes back this direction, then it emits light. It lights a wave, and these are associated with various photons of various energies, which we call, at least in the little slice of the electromagnetic spectrum we can see, red, orange, yellow, green, blue, indigo, or violet. And so that energy comes out in the form of light for us, and various colors. So if you ever put a chemical in the flame and you saw orange, or you saw red, or you saw green, green's one of my favorite ones, then what you're seeing is the fact that that electron was promoted to this higher energy level, and then when it came back down to ground state, it put out that energy in the form of light that we could see. So the chemical behavior of an atom is determined by the distribution of the electrons and the electron energy levels and sublevels, and the periodic table of the elements shows the electron distribution for each element. I like to tell students to think of it as a giant bingo card, because again, if you've got the general idea here about the metals are on the left, the nonmetals are on the right. And I like the classrooms that have the periodic tables where they've color-coded solid versus liquid versus gas. And there's very few liquids and more gases than liquids, but still both are pale in comparison to the abundance of the amount of solids. All right, so we have our shells here. And again, I'm going to keep harping on it, but this shell thing is very problematic. But when we look at the periodic table, it is arranged based on reactivities, which is based on electron arrangement. So the first shell, I die a little each time I say it, is the 1s, or the first energy level, I guess is how they're doing it. No, they're not. They're doing it by sublevels. Okay, I had to look at it for a second down here on the third shell. All right, so that's going to be my 1s, and that's going to have one electron in it. So you may have seen that notation in chemistry, where this would be 1s2. And then we start the second shell, which is we fill up the first shell. It only has room for 2 electrons. So any s only has room for 2. So when we move out into the p's, we can put 6. But again, they repel each other. So there's one, and one way over here, and then a third. And they're going to repel that way and fill singly for half of the orbitals until they have to start to pair up. And so this is going to be, it's still it's going to be S2 because that's full, but now it's P1 and this is S2, P2. No one's going to ask you this on the test. I'm just relating this back to something that you probably already know. And so you may have even drawn the p orbitals like this and said, here's electron 1, 2, and 3. Because electrons repel each other, they want their own room, much like brothers and sisters, or especially a pair of sisters. And then they won't put another electron into this orbital until we get to the 4th. and they have to be of opposite spin, up arrow indicating spinning one direction and down arrow the opposite direction, because they also set off little magnetic fields. But we won't get into that. So what do you need to know? Well, this is the stuff you probably already know. That the valence electrons are those in the outermost energy level. So valence electrons live in valence sublevels. And then the chemical behavior is most determined by these valence electrons. Element with a full valence are chemically inert, and those are our noble gases. I am so old that I actually learned in high school they were called inert gases until some jerk came along and made a compound with xenon. And he made xenon tetrafluoride, 4 fluorines attached to it. And then we couldn't call them inert anymore, because inert means they don't do anything, they don't react. So they became noble gases. All right, so we talked about this, just a blow-up picture of what we just said. So here come some more questions. You ready? Read the question. I'll read it to you. And then pause me. Whether an atom will be able to interact with other atoms can be determined by looking at the ratio of the protons to the neutrons in the nucleus, whether it has an even or odd number of electrons, determining the stability of the electrons in their valence orbitals around the nucleus, or identifying the atom as a metal or a nonmetal. All right, pause. Think about it. No cheating. All right. Looking at the ratio of protons to neutrons is going to help us determine whether or not it's a stable isotope. Is it radioactive or not, in other words? Whether it has an even or odd number of electrons is just a silly answer. It doesn't really matter about anything. Determining the stability of the electrons in their valence orbitals around the nucleus has a lot to do with whether they will interact with other atoms. We just talked about those in those noble gases. Identifying the atom as a metal or nonmetal? No, because the properties on the periodic table repeat. And so you're going to, as you go down the table, you're going to have a real big swing in what the numbers are. So just looking at it as to what their number is, that is not going to help you at all. All right, so an orbital is that I keep using is a three-dimensional space where an electron is found 90+% of the time. Each of these electron energy levels consists of a specific number of orbitals. Now, again, this is not something they're going to test you on the AP exam. I'm just trying to explain to you how this works, because you need to know for college. Whether you're in a chemistry class, a physics class, or a biology class, this is fundamental knowledge stuff. So you probably have already had it, and if so, happily fast forward through this. But we have 1s, 2s, and 2p. So essentially, you got to think about the fact that we have 1, 2, 3, 4, 5, 6, 7 periods on the periodic table. So if you take a naked periodic table, I always tell students, and especially in chemistry, just number it 1, 2, 3, 4, 5, 6, 7 down the left-hand side. Then you're going to have S in the first, S and P in the second, S and P and D in the third, and S and P and D and F in the fourth. And that goes all the way down. And the fifth could have G, beyond F, it could have G, H, or I. I know what you're thinking. What the heck? Why didn't we just do A, B, C, D? Well, because back in the day, 100 years ago, we learned all of this through something called spectroscopy. Spectroscopy, like microscopy, but spectroscopy. And spectroscopy had words to describe the spectrum. Sharp, principal, a principal line, I had to think about that one a second, diffuse, or fundamental. So they used those words that they had used in describing the spectra to do that. And they thought that was the end of the story. They thought F was the be-all and end-all of all the sublevels until much later, and they got better technology and discovered there were more, and they were out of those descriptive words, and they kind of went, oh, well, we'll just go alphabetical from there, at least in the English alphabet. So F, G, H, and I. So it goes on out. S, P, D, F, G, S, P, D, F, G, H, S, P, D, F, G, H, I. So there are many more. Now, none of these are ever occupied unless you're doing experiments and you're really perturbing electrons, and it's all in a lab. Nothing in nature happens that way. So again, I want to get rid of this idea of a shell. This is like just a fuzzy region where the electrons can be in a three-dimensional world. So it's more like if you blew up a balloon. It's more like that. And the 1s orbital and the 2s orbital, they just look like spheres. Just a sphere of area where those electrons can zip around in. When you get to the p's, life is a little more interesting. They're more like dumbbells. And so here's one figure 8 looking thing, like a dumbbell, as in I'm lifting weights, I'm lifting a dumbbell. So this one we would probably say is on the z-axis, so you've got to imagine it coming in and out of the screen as you see it now. That lobe you would see at the front, you'd have to flip your paper over or your computer over to see the back. And then another, let's see if I can change colors of my pen here. See if I can get all fancy. Another, they've got the x-axis in this diagram coming in and out of the screen, but you get the idea. So that's another. And then, what do you want to go with? Let's go with purple. The last one. So these are 3 different regions of space. So what I like to do in class to kind of explain this is I like to get 6 balloons in, have 3 pairs of the same color of balloon, and do just what I did with this little drawing. But I have students blow them up, tie them off, and then I tie 2 balloons together that are the same color. And you can actually construct this in a 3-dimensional plane. So you can see that one of them lies on the x, one of these little dumbbell regions lies on the x, one lies on the y, the other lies on the z. So the fact that we have 3 orbitals and a pair of electrons can fit in each one means we get 6 electrons. They have to be of opposite spin in the same orbital. And then see if I can do this. There we go. So that gives 6 regions of space for these to exist. Again, nobody's going to test you on the biology exam, but it's overlay them and you start getting this overlay. So these are the regions of space where those electrons can move. Just trying to connect something that you might have already known. All right, so now the formation and function of molecules depend on chemical bonding between atoms. So I'm going to give this a little quick intro here, and then we're going to stop this recording, and we're going to actually, because this is a good breaking point, do a separate recording on this where I'll recap a little bit of this. So atoms with complete valence shell can share or transfer valence electrons with certain other atoms. You probably already know a word for that, and you probably already know a word for that. If they share, then we use the word covalent, co meaning a cooperative term, they're sharing, versus transfer, which is like stealing, and we call that ionic. So these interactions result in atoms staying close together. So if they're sharing, they have to be close together. If they're stealing, they have to be close together. And they're held together by attractions of chemical bonds. They are electrostatic attractions of varying degrees, strengths, if you will. Okay, so covalent bonds versus ionic bonds. The covalent bond, co meaning cooperate again, is that sharing, like sharing, you have to cooperate with someone to share, of a valence pair of electrons between 2 atoms. In this covalence bond, the shared electrons count as part of each atom's valence shell. That confused me when I was in high school. And I remember vividly going up to my high school chemistry teacher and saying, I don't get it. How can I count these electrons twice? It feels like cheating. And he says, if I ask your mother, how many kids are in your family, what's the answer? I would have said 1, but I'll make it up and say 3. 3. If I asked your mother, what would she say? Also 3. They share you. They share the children. So think of it that way, because that statement confused me because I'm simple-minded when I was in high school. So indeed, they are sharing a pair of electrons. If one pair happen to be shared, we get a single bond. Looks like that. So if I have a single bond between carbon atoms, it's going to look like that when we draw it out. If you have 2 pairs being shared, then it's a double bond, and it would look like that. If you have 3 pairs being shared, then it's a triple bond. and looks like that. Now, we'd have carbon's going to make 4 bonds, so I'm going to put in the rest. I'll angle them that way. That would probably be a better plan. Carbon always has 4 bonds, no more, no less. Furthermore, not every element on the planet can do this action, only these elements, which we say C-nops, like Cyclops, if you've read the Odyssey. You know, that the mythical monster with one eyeball in its head, a Cyclops. So it sounds like Cyclops, but it's C-nops. Only carbon, nitrogen, oxygen, phosphorus, and sulfur can do this, and they can only do it with each other as well. So I can have that, but I can't have this. Okay, so when I say they can double bond, not only are they the only ones that can double bond at all, furthermore, they can only double bond with each other. All right, I'm going to stop this recording here. I'm going to pick up with a little bit of this and maybe redo this a little bit because you're probably going to sleep between now and the next time I hear from you, or you hear from me, I guess is a better way to put it. And do come back, though, because the next portion of the chapter is even more important than the first portion. Until later, I hope you have a great rest of your day.